So just a some brief words about SynBio, or maybe about the history of why we're here, I guess. So in 2019, or actually in 2018, we kind of formed the first iGEM team of the University of Zurich. The iGEM competition has been going on for 15 years, I think, or even more. And we've kind of started it here at the uni. And then this year, there's been the second team ever participating. And now, I guess most of you or some of you will be part of the third team. And SynBio is a student association that we created kind of along with the first iGEM team to kind of make sure that it's going to continue and that iGEM is going to be supported along its way, and that it's as easy for you to set up as possible every year. So before I get into what exactly iGEM is, we kind of need to understand what synthetic biology is, because iGEM is the world's biggest competition about synthetic biology. So, but before that, just briefly to who I am and why I'm standing here, I guess. So I graduated with my bachelor's just like 3 months ago. I was part of the first iGEM team together with Lynn over there, first row, second row, and we had 2 other team members who are not here today. And then I'm also part of the SynBio board, which is the Vorstand. So if you have any questions about SynBio, I would be the right person to ask later on. All right, so synthetic biology. How many of you kind of know a bit about synthetic biology? Can you raise your hands? Okay, yeah, you should. You guys should. Okay, good. So synthetic biology is a fairly new field in biology as a whole, and it combines a whole different disciplines like genetics, molecular biology. But the most important thing is that it kind of tries to take this viewpoint to see cells as kind of machines, because synthetic biology incorporates biology as well as engineering. And what we basically try to do is, if you look at a cell with all its components, the mitochondria, for example, we kind of try to look at them like a sort of, factory or compound that does a specific thing. Just like in an iPhone, you have a processor or, a graphics card in a computer. And that's basically the main point of synthetic biology, to view cells as kind of machines with interchangeable parts. And if we understand them well enough, we could potentially, change them either through something called, metabolic engineering, where you try to change how a cell basically lives, how it functions on a molecular level, in kind of the compounds it produces. You could change the structure, you could change the cell membrane, you could change maybe the nucleus of a human cell, and you could maybe even build them from scratch. And actually people have designed synthetic genomes, which basically means you write a bunch of code that makes sense, and then you synthesize all the DNA. And with that, they already created kind of the first basic life form, where they completely synthesized the genome and created a living organism from that. So that is synthetic biology, and it is so much more, but that is kind of the rough outline. Now, just to give you a few more examples to tell you why I and we think this is really, really important. If you think how drugs are made, for example, pharmaceuticals or even things like insulin, usually they're produced through chemical processes. Well, insulin actually not anymore. Insulin was the first compound that we went from extracting it from pig stomachs to taking the human insulin gene, putting it into bacteria, and now they're producing all the insulin. No pigs needed anymore, and for a much cheaper price as well. And complex drugs are manufactured in chemical processes, but if you could just take maybe a couple of genes, throw them in some yeast, as you can see on the picture on the left, and the yeast could produce the compound for you, could save so much money and material, and also manpower in creating drugs that could help people. Toxin detection is actually a very, very good example, because that's what this year's team is trying to do. So the figure on the right, you know, if you don't quite get what's going on, that's fine. The basic, it's important that, you know, you have a certain molecule, which in this case is called the trigger RNA, and this could be from a toxic bacteria. And if you have, you know, an organism engineered to kind of recognize this, it activates some process in it that maybe changes the color or produces a fluorescent light. So basically, you can take some water, add, you know, a drop of bacteria liquid to it, and then it can change the color based on whether or not there are specific toxins in it. And you can imagine how important that could be for things like cholera in Africa, you know, because that's still a very widespread problem. And, you know, the list goes on. We have also projects that can be like, you know, in terms of energy conversion to produce hydrogen, to produce energy just with bacteria. And that's kind of really what synthetic biology aims to be. And you realize it's not really a form of basic research, it's very, very much applied. And that's really important to keep in mind. And that's, I guess, why we also like it so much, because it's focused on the real world and what we can actually change. All right, and you know, iGEM is then, or kind of follows really that principle, as in it's a student competition concerned with creating your own project about synthetic biology. And at the start, you always think of a problem that you want to solve. For example, a drug that's super expensive to manufacture or not accessible, and you want to make this process easier. And in iGEM, you as students can do that, because it's not about people with a PhD or professors who take on these projects, but it's really students from, you know, bachelor level to masters as well. So it's really about you, and that's the great thing about iGEM, because you can do something that you wouldn't otherwise be able to do, which is gain experience in a lab or working with a team to really make your own project as you want it. The competition lasts from January to November. I'll show you a quick slide afterwards in terms of timeline. I'm good. And yes, I mean, really, it's your project from the start to finish, I guess. No. Okay. So this is kind of the timeline of how an iGEM project works, and I'll just briefly touch upon this. But as you notice, we're now in October. So the current iGEM team is at the end of the competition, mostly. Well, we still have some deadline, I know, but and so we're recruiting now to just make it easier, that you're not going to get into stress. You know, when it's already April and you don't have or May and you don't have a project yet, things might get stressful. Okay, sounds good. Yeah, and you know, at the end, which I highlighted, the giant jamboree, that's kind of the end of iGEM always, and it's it's a giant competition, it's a giant meetup basically of all the teams that participate. And we went there last year to Boston. It's always in Boston. This year will be the last year that is in Boston, but it's not, because, you know, Corona. But starting next year, we'll actually be in Paris. So, and it's a bit difficult to explain how the giant jamboree is, but I figured I'd just show you a video of how vast this actually is. So, 350 teams and a lot of students from all over the world. Yeah. That was 2019. That was when me and Lynn were actually there. And that's a really, really great memory. Yes, we saw that. Okay, just very briefly at the end, near before the end, you know, I told you a bit of ideas of what synthetic biology can do, but in terms of projects, you know, you're maybe a bit limited by the lab that hosts you in the end, but this you can, you know, on your own choose to a certain extent. But you know, the project can be it can be many things. So for example, the first year, it was a very research-focused, lab-focused project. But you can also focus your project more on the human interaction, you know, how the product or the thing that you're trying to create would impact society. So it can be more of let's say, policy or society-oriented project as well. And you're not very limited by, you know, just doing lab research. You can do so many things with iGEM. You can do a complete computational project, not being in the lab once, if that's what you want. I know some people who might want that. Or it can be very entrepreneurial and really trying to create a project that you could actually then market. So a lot of iGEM projects have actually gone into a startup and they're now selling what they created as students, which is kind of amazing. Now at the end, you know, I kind of, my job basically is to get you excited for iGEM. So I just want to give you some more reasons of why you should do it. And you know, of course, it's an amazing learning experience. You learn so many things that you will not learn at the university course, because you will be working on your own project for a whole year, and you don't get that at the uni otherwise, pretty much. You get international exposure, of course, because you meet so many different people and you talk to a lot of different teams, and that's also great for your own development if you work in the team for such a long time. And you will certainly become experts in synthetic biology as well over the time. And you know, it's a lot of fun. It's really great. It's hard work, but I think at the end, it was really worth it. And you know, if you're not sure whether or not you have the experience or whether you're ready, I would just like you to consider that usually it's the things that you regret are usually things that you don't do. So speaking from my experience, I really encourage you to do it and try to not regret it if you don't do it. So that's my part. I'll be handing over to Lynn now, if that's correct. You'll have opportunities to ask questions later for, you know, the 2019 team, 2020 team, and also for SynBio. If you have questions about SynBio, about our organization, just come to me.